Now we come to the juicy part, the clinical significance of nucleoside. And so great, now we absolutely understand where the nucleoside came from, right? And we talked about them. We said we have adenosine, guanosine, cytidine, thymidine, uracil. Why do we care? Why do we care about the nucleosides? In fact, they're extremely important, all of them. Let's start with the most important one, which is adenosine, right? So, clinical significance of adenosine or deoxyadenosine, same thing. So, point number one. Adenosine in adenosine deaminase deficiency. Now remember, we said adenosine was simply a ribose sugar, right? That is connected to a nitrogenous base, okay? And this nitrogenous base has an amino group. Remember, 2 rings, 1 ring and 2 ring. Sorry for the way I draw them, but anyway, okay? There's an enzyme. This enzyme is called adenosine deaminase. What it does is, look at the word. Remember, adenosine, so basically it's working with something, which is the adenosine, deaminase, so it's removing the amino group, aminase, right? So it removes this group. Now, if we remove this group here, okay, probably shouldn't have done that, but okay, here, there you go. Right? So if it removes this, now this structure over here is no longer called adenosine. Instead, it's called adenosine, right? Adenosine, basically, it goes through this pathway. It's called a purine salvage or a purine degradation pathway. The adenosine becomes something called hypoxanthine. Here, hypoxanthine, and then the hypoxanthine becomes something called xanthine, right? And the xanthine, eventually, it becomes uric acid. And uric acid is secreted out of the body by your urine. Now, if you're making too much uric acid or you're under-secreting uric acid, you get a condition called gout, right? And that's where, basically a lot of the patients, it's very painful, right? They say, oh, my toe, it's killing me, it's hurting, it's inflamed, whatever. The purine degradation pathway is not the reason we're talking about this, however. There is a condition, and this condition is called adenosine deaminase deficiency. Basically, this enzyme, imagine a patient has very, very low levels of it. And so, okay, and since there's low levels of this enzyme, that means that adenosine, the amino group on the adenosine, is never going to get removed, right? So what? Let it be, right? That's what you might think. And where's the big deal? So let's have the adenosine just floating around and everything's going to be great. Well, there is a problem. Once adenosine starts to accumulate, it's going to give us a very high amount of adenosine and something called DTAP. Oh, sorry, actually, not DATP, sorry. It's like a mental block at this stage. DATP, deoxyadenosine triphosphate. So basically you have a very high level of adenosine and deoxyadenosine triphosphate. Don't worry about this name, I'll explain it in a moment, okay? But anyway, now once they start to accumulate, they become very toxic to the cells, and especially lymphocytes. Lymphocytes are one of the groups of the white blood cells. They help us fight off infections, correct? So if they're toxic to lymphocytes, they're going to start killing the lymphocytes. Lymphocytes are going to start to die. And that means that the patient is going to become susceptible to having recurrent infections, okay? And this leads to a disease called SCID. SCID simply refers to severe combined immunodeficiency. Well, one of the reasons would be this. It can be due to other reasons, by the way. SCID is like responsible for about 10 to 15%, or sorry, the adenosine deaminase deficiency is only responsible for 10 to 15% of the cases of SCID. A lot of them are actually X-linked recessive, but we'll talk about it when we reach immunology. By the way, academia loves severe combined immunodeficiency because it was the first disease to actually be cured by gene therapy, right? Now, so this is the first clinical significance of severe combined immunodeficiency. Number 2. So its role in cardiology, right? Red for the heart. Adenosine has 2 major roles. It's actually used as a pharmacological agent in some cardiac conditions. Number 1 is something called SVT, supraventricular tachycardia. What happens in supraventricular tachycardia is that the heart rate is going to be blowing through the roof, right? The patient comes in, usually the patients have had this episode before, okay? So they're kind of expecting what's gonna happen. One of the things that the patients with SVT are always complaining about is something called an impending death. They feel that they're gonna die because that's what SVT, that's what it feels like. It feels like death, okay? So he comes to the emergency room, or she comes to the emergency room, and she's like, "Doc, please, I'm gonna die, etc. I'm just gonna throw myself on the bed over there. You get your adenosine and give it to me, and we'll forget that this ever happened." Right? By the way, doctors are the most thanked and the most sued people on the earth. So, Anyway, SVT, you give the patient adenosine. Why do you give him adenosine? Because once you inject adenosine into the patient's arm, it's going to cause a decrease in something called the AV conduction. Again, listen, I know you probably don't understand a lot of this. Maybe you're a first-year medical student. Maybe you're pre-med, whatever the case is. All I want you to know is that adenosine, we use it a lot. It has a lot of clinical significance. So we can use it in supraventricular tachycardia. Where else can we use it? We use it in a pharmacological stress test, right? What's a stress test? A stress test is in the cardiologist's office. They usually have a treadmill. They put the ECG on the patient, and then they ask the patient to walk to see if there's any areas of ischemia, meaning that there's a decrease in blood flow to that area. And so he gets up and he starts to walk, and the cardiologist would look at the ECG and determine if there's ischemia or if there's unstable angina or whatever, right? Now, what if the patient comes in, he's crippled, or he's an older patient that has maybe osteoarthritis, right? Any reason that makes maybe has a broken leg, right? So how am I going to do a stress test? Well, I give him adenosine. Why do I give him adenosine? Because adenosine is a vasodilator. That means that the vessels, especially those around the heart, when I give adenosine, they're going to dilate, they're going to expand. So, it becomes like this. It expands it, right? Once we talk about pharmacology, I'll explain it in detail. For our purposes right now, just know adenosine is used as a drug in stress test. Okay? Point number 4. Okay, so first of all, apnea of the newborn, right? Or what happens is that some premature babies, right? In premature babies, the respiratory centers and brainstem, they're not really well developed, right? And adenosine is kind of, it likes to be calm, it likes to relax the body, etc. And so one of the things it does is, it causes a depression in respiration. So what do? We give the babies caffeine. Please never give a baby a coffee. You're gonna kill him. We give it in a form called caffeine citrulline, I think, right? Basically, it's in a very narrow therapeutic dose. What happens is, oh yeah, listen. Now, if you've ever played Elden Rings, there is something called the mimic, right? Basically, what a mimic is something that looks exactly like you. It functions like you. It talks like you. It walks like you, right? A mimic. Caffeine is a molecular mimic at a molecular level, at a very small level. It's a mimic to adenosine. Adenosine binds to receptors in the brainstem, the respiratory center, and causes depression of respiration. That's why when we are asleep, the breathing quality, it kind of decreases, it becomes more shallow, it becomes slower. And in babies, in premature babies, where they have apnea means they're like, they can't really, the diaphragmatic contractility, the respiratory centers are depressed, basically. So we give them caffeine. Caffeine, since it looks like adenosine, is going to have a fight with adenosine. Suppose that this is a receptor for adenosine, something that engulfs adenosine to make adenosine give its action. What happens is, Mr. Caffeine is going to come in and push out adenosine, get lost adenosine, and it's going to sit eventually in his place and causes stimulation of the respiratory centers. And so it increases the diaphragmatic contractility, it increases the respiratory rate, and the babies have less apneic episodes and they get better oxygenation. Most of you, probably all of you, drink coffee, right? So why does coffee keep you awake? Again, what happens is, you see, again, we said that adenosine itself is one of those molecules that binds in specific areas in the brain, and it causes a sense of calmness, drowsiness. It makes you want to sleep. Now, caffeine looks like adenosine. And when you drink enough caffeine, it's going to compete with adenosine for the receptors. And so it pushes away the adenosine and sits in the receptors, and now you're awake and you're alert and you're watching Draminor Medicals and you're happy. The thing about adenosine is adenosine never goes away. While caffeine is sitting there in its receptors, adenosine is just accumulating in the background, right? It's watching, it's waiting, because eventually adenosine knows that caffeine is going to go bye-bye. It has the half-life of caffeine is maybe like, 8 hours, 5 hours, 6 hours, something like that. So after 5 to 6 hours, and you're alert and you're happy, suddenly caffeine goes bye-bye, and adenosine, the huge amounts of adenosine that have been accumulating in the background, rush into the receptors, and now you get what is called a caffeine crash. And that's why you get a caffeine crash, that's why caffeine keeps you awake, and that's all. Okay, and that was the clinical significance of adenosine. Okay, let's look at guanosine. Okay, so guanosine analogs, right? We're going to use the green color because, it kind of fits the theme. Guanosine, right? Now, this is biochemistry, great. We talked about, in adenosine, we talked about the immunology, we talked about cardiology, we talked about pediatrics, we talked about sleep medicine, right? Now we're talking about pharmacology and virology. We're going to see how exactly we use guanosine analogs to kill viruses. Number one, so guanosine analogs, again, there are going to be acyclovir, it's going to be ganciclovir and ribavirin. Let's start with acyclovir. Now, to understand how acyclovir actually works, let's look at the normal structure of guanosine, right? Or deoxyguanosine. Because acyclovir, usually we give it to patients that have herpes simplex virus, right? So, this is the ribose sugar. Again, this is going to be a deoxyguanosine, right? Here's the 3 prime, here's the 2 prime, and attached to it is going to be a guanine nitrogen base, right? So 1 ring, 2 ring, and here's an oxygen. There's no amino group because that would make it adenosine, right? Now, this is the normal structure of guanosine. Acyclovir, you know what, I'm not even going to draw it anymore. Listen, acyclovir, you see this 3 prime here? This is what acyclovir is. It's the exact same structure, but without the hydroxyl group. Now, if you recall, if you remember, we said that when the nucleotides, remember I said I was a nucleotide, and once, or I am a sugar, and I grab a nitrogen base, and I grab a phosphate, and now I'm a nucleotide. And the way I build myself is, let's say that this is the 5 prime end, my feet are the 3 prime end, another one of like nucleotide, whether it be adenine or guanine or whatever, they come and they stick themselves underneath my feet because it's the 3 prime end. Once acyclovir enters the cell, here's a big fat cell for you, okay? And here is acyclovir, and so acyclovir enters the cell. Now inside the cell, what's going to happen is that the virus that's inside the cell, here he is, he's looking all mad, and he's really bad virus. So he's going to take the guanosine and he's going to put a phosphate on it, right? Or the acyclovir, sorry. Of course, the acyclovir is nothing but a guanosine with a missing hydroxyl group on the 3 prime. And so he's building his DNA. So far, let's say he built, he put an adenine, then he put, a thymidine, and now he went and he put, or he thinks it's a guanosine, but in fact, it's going to be acyclovir. So it's like a fake guanosine, right? And he's happy. He attached this to the 3 prime. He attached this to the 3 prime. Now he goes and he takes, let's say he takes, what does he take? Let's say he takes thymine again, and he wants to attach it here. But behold, it has no 3 prime, or it has no hydroxyl group on the 3 prime. And so what do we get? We get chain termination, DNA synthesis halts, it stops, and you can imagine this happening on thousands and thousands and thousands of cells that contain this bad boy. Eventually, he won't be able to replicate himself, and he dies. The patient gets better. That's acyclovir. Okay? Now, what's the other drug? Ganciclovir, right? Now, I'm not gonna redraw anything here. I'm just gonna move acyclovir and this, and I'm gonna put ganciclovir, right? Why? Because ganciclovir works on a virus called cytomegalovirus. Basically, cytomegalovirus is more resistant to acyclovir, and so, they developed a new drug that is called ganciclovir, which has more potent activity against the cytomegalovirus. Now, ganciclovir, it in fact does have a hydroxyl group. The only difference is the hydroxyl group is shaped kind of funky. It's weird the way it's shaped. So it doesn't really cause immediate halting of the DNA synthesis. However, it kind of slows it down. Eventually, if enough of the ganciclovir come in, they are going to cause halting in DNA synthesis. Okay? A very important point, by the way. Listen. About acyclovir and ganciclovir. About both of them, in fact. Listen. The main difference, and ganciclovir causes severe side effects. One of them is neutropenia, which means it causes the white blood cell levels in the body to go down. Acyclovir doesn't do that. Why? Acyclovir, the only thing that can activate it when it's inside the cell is the viral kinase. Okay? And therefore, if a cell is not infected with the virus, even if the acyclovir comes and enters it, it's never going to be activated. There's no phosphate that's going to be put on it, and so it's not going to work. Ganciclovir, on the other hand, it can be activated by the virus, by a cell that's infected by the virus, or even if it enters a normal cell, it can be activated in there. And therefore, you would imagine that it can in fact affect cells that are infected with the virus and cells that are totally normal and totally healthy. Okay? Okay, so cytosine's up next. Let's get rid of the guanosine. Let's leave the cell, maybe we'll need it, right? So, the clinical significance of cytosine. The first drug here is something called cytarabine. So what is exactly cytarabine, right? Now let's make the cytosine, right? Again, 1', 2', 3', a hydroxyl group. Everything's nice, right? And of course, the nitrogen base, cytidine, here, with the amino group. And forget about that. Cytarabine is used in leukemias and in lymphomas, but mostly in acute myeloid leukemia. Why? Well, cytarabine targets the tissues that are rapidly dividing. And in leukemias, in any cancer, cancers are rapidly dividing as well. So how does it exactly work? How does this nucleotide work? Basically, listen, cytarabine, it looks like azacitidine. However, the sugar that is used is an arabinose sugar. It's a pentose sugar, remember 5 carbons, but the structure of it is kind of different, right? For instance, first of all, it has a hydroxyl group on 2'. Okay, that's no problem. I mean, sure, it makes the DNA less stable, but what it actually does is, consider this is the cytarabine, right? And so it has the 2' hydroxyl group, 3' hydroxyl group. And then, fuck, I'm tired, I am. I'm really tired, sorry. Okay, anyway. So, the way cytarabine is done is that the hydroxyl group on the 2' end is going to be oriented in a manner that is opposite to the 3' end. So basically, now you have a hydroxyl group here and a hydroxyl group here on the 3' end, but look at the hydroxyl group, how it's oriented on the 2' end, right? And that means when the tumor cell, when the cancer cell incorporates it into its DNA, it puts cytarabine, and now it wants to go and bring another nucleotide, which is this one. Look, it's oriented, it's normal, right? And it wants to put it and stack them on top of one another. Look what happens. The mere presence, the orientation of the hydroxyl group on the 2' end of the cytarabine will not allow it, the geometry, let's say, the 3D shape of it, is not going to allow the incoming nucleotide to bind to it. So what happens? Chain termination. The problem with cytarabine and drugs like it is that since they target rapidly dividing cells, that means that they're going to target also cells that, are present in the bone marrow. And therefore, they're going to also affect the number of white blood cells. They're going to start to die. Remember, there is DNA synthesis termination. Platelet levels are going to decrease. Patients get something called mucositis. Basically, it's a gut inflammation. Why? Because the cells in the small intestines and the large intestine, they're always, turning over. They're always changing. They're rapidly dividing. Therefore, these patients, they get diarrhea, they get vomiting, etc. Low white blood cells, low platelet levels. Okay, now that was cytarabine. Number 2. Okay, so azacitidine and decitabine. Listen, very easy. Again, basically, you know what? First of all, you've got to understand something. There are some genes. Like suppose this is a fragment of a DNA, right? And it contains genes. And these genes are going to be maybe rich in cytidine monophosphate, right? So CC, for instance, GAC, whatever. These genes, one of the important functions they have is that they are they're called tumor suppressing genes, right? So what does tumor suppressing mean genes? What does tumor suppressing genes mean, right? Basically, a tumor suppressing gene is a gene that makes proteins eventually that go and fight cancer cells. They do not allow the cancer cells to live and replicate, right? Now, why am I bringing this up here? There's an enzyme. This enzyme is called the DNA methyl transferase, or for short DNMT, right? So look at the name, DNA methyl transferase. What does it do, this enzyme? Basically, it takes methyl group, right? Remember, we said the methyl group is CH3, and it transfers it. Where does it transfer the methyl group? It takes the methyl group and puts it on the 5 carbon end, or the 5 prime of the cytidine, right? And you can imagine when this happens here, this process of methylating the cytidine or the cytosine causes it to become silent, meaning that if there is a tumor cell and it's roaming around and it's like, yeah, I'm gonna kill this man, this woman, this boy, this girl, whatever. I'm gonna kill this body. And so since the tumor suppressing gene, this gene here, is silence, all it can do is just watch it while it goes and kills the body. Now, this is where azacitidine and decitabine come in. What they do here is these are cytosine analogs. What does that mean? It means, listen. It means they look exactly like cytosine, right? The only difference is at the 5 prime carbon here, at the 5 prime end, instead of a carbon being here, they put a nitrogen base. So the drugs, what they do is we manufacture it in a way where over here at the 5 prime end, there's no carbon, instead there's a nitrogen. Okay, so what? So what? I'll tell you so what. So when the DNA methyl transferase comes and tries to put a methyl group right here, it cannot. It's like, well, where's the carbon? And azacitidine and decitabine, they're like, well, no carbon for you here, boy. It's only nitrogen. And so now DNA methylation, it's just looking there. It has its methyl group, and it's grabbing it. So, what do I do with you? We can't. Anyway, the end result is what? The end result is, since there is no methylation, that means that there is going to be no suppression of the tumor suppressing gene. This is not going to be suppressed, and therefore it's going to be active. And now, the gene itself, it pops up its muscles and starts fighting tumor cells. That's the idea behind azacitidine and decitabine. Very simple, right? Beautiful, beautiful. And by the way, don't worry about the tumor suppressing genes and all of that. We're going to discuss it in detail. We have a lecture about it. Just trust the process, okay?